So in this video, I'm going to be talking about meiosis. So meiosis helps heritable information being passed down, and this heritable information provides for the continuity of life. Now this cell is the first step of meiosis, which is in step G1, and it has 4 individual chromosomes, which has been passed down. The pink ones represent the chromosomes from the maternal, and the green ones represent the paternal. Now, this cell is now entering synthesis, where these chromosomes become duplicated. And when they become duplicated, that means that they are duplicating their DNA, and it develops sister chromatids. And so now this is the third step of meiosis, and this step demonstrates crossing over. So these 2 sister chromatids in the middle right here, they cross over in this little area right here, and it exchanges the DNA. So in the next diagram of the cell, as you can see right here, now these sister chromatids have exchanged DNA, and you can see the different colors. This pink duplicated chromosome now has a green part, which is DNA from this duplicated chromosome right here. So this step can also represent crossing over, which is very vital to life because it creates genetic variation. And they are also moving to the middle of the cell, which is now separated at step 5. And these homologous duplicated chromosomes are now pulled apart from each other and ready to enter M phase, which is where mitosis can occur, and it's the division of the nucleus, which is this step right here. And this step also is demonstrating cytokinesis, which is division of the cytoplasm, and they now separate into 2 different cells. So now, as there are 2 different cells, we are entering in meiosis 2, which these 2 haploid cells now have duplicated chromosomes, and they have already gone through crossing over, and therefore can go into step 8, which is moving to the middle of the cell in order to get ready for M phase. And so at step 9, they are being pulled apart and at random selection. So the separation of the homologous chromosomes ensure that each gamete receives a haploid set of chromosomes composed of both maternal and paternal chromosomes. So now, as the sister chromatids are split apart in step 9, they can now go through cytokinesis once again at step 10 and can now create the gametes. So one of the main purposes of meiosis is to ensure genetic diversity, which means that each gamete receives 1 complete haploid set of chromosomes, 1 from maternal and 1 from paternal. So fertilization involves the fusion of 2 gametes, which is the number 1 reason of increasing genetic variation. Crossing over was another reason to create genetic variation. So genetic variation in populations by providing for new combinations of genetic information in the zygote. And now we are back to step 1. And restores the diploid number of chromosomes, which is 23 from maternal and 23 chromosomes from your paternal, from your mom and your dad.